Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is the influence of storage and transport on the microflora of a raw milk. Okay, the microflora of the raw milk are already discussed in a previous lecture, and the topics related to this are already uploaded. Now here what should we are going to see? We are going to see that what is the impact on this microflora of raw milk when it is stored or transport. Okay, so during the transportation and the storage of the raw milk, what is the effect of their microflora, what is the changes of the microflora, we are going to discuss in a today's lecture. We all know that this microflora are of two types. One is harmful and one is beneficial. Okay, so this both type of micro organisms are present in a raw milk. Okay, so here what should we are going to see? We are going to see that what is the impact of the storage and transportation, what is the impact of the storage and transport of the raw milk regarding the harmful bacteria and regarding the beneficial bacteria. Okay, so we all know that when milking is done, then this milk are what, this milk are stored in a tank, and that tank are called a bulk tank. Okay, so that to await a collection and subsequent delivery to a collection center. Okay, suppose that if this is our main industry. Okay, but before reaching this main industry, this is your industry, milk industry, dairy industry. Okay, this is a subunit where the milk are stored. Okay, so before reaching this, the milk are stored here. So what is the storage criteria and how they transport to the industry? What is the transportation impact? All this we are going to discuss in the today's lecture. After the production, milk usually stored in a bulk tank to await a collection and subsequent delivery to a collection center or a processing plant. The raw milk is delivered to the processing facility in a metal milk can or a insulated fiberglass container in 100 lb quantities or in insulated horizontal tankers in a bulk quantities. Okay, raw milk, when milking is done, then after that it reaches to the processing tank in a metal milk tank. Okay, or in a insulated fiberglass which contain around 100 lb quantities, or in insulated horizontal tanker in a bulk quantity. Okay, if milk said to be a bulk quantity, then it should be sent by a insulated horizontal tanker. Okay, you can see the tanker like this. Okay, so here what it is made up of the insulated tank, and it is called a insulated horizontal tanker. In most developed countries, construction standards for bulk tanker require that milk contact surface of the tanker be fabricated from a stainless steel with a specific polish specification for a cleanability. Okay, if we talk about the developed country, then the construction standard for a bulk tankers. Okay, so for a bulk tankers, there is a standard for their construction. Their surface should be smooth, and it is made up of fabricated from stainless steel, and it is polished with a specification for a cleanability. Okay, another requirement in that tanker be insulated sufficient to maintain over a 24 period the milk temperature to within a 1 degree Celsius from the temperature at time of pickup. Okay, so one requirement of the tanker is that it maintain the temperature. Okay, so the temperature of the tanker should be around 1 degree Celsius for a 24 hours. It should be there. Okay, the otherwise the microflora of the milk will be changed. Now the next one is your can collection. Okay, so the first storage or a transportation is done with the help of this can. Okay, in countries with emerging economic, raw milk are usually is collected in a metal can. Okay, so in a countries with the emerging economic, usually this raw milk are collected in a metal can, and then after that it is transferred to the collection center within a 1 day after a milking. Suppose that if a milking is done in a Monday, okay, then it should be reaches to the milk dairy industry or a milking center Monday. Okay. It is stored in a collection center, then it reaches within a 1 day after milking. Sorry, it will reaches to Tuesday. It will reaches to Tuesday. So for a whole day, it is stored in a collection center, and in a collection center the temperature and the storage condition should be maintained. Here the milking is cooled and placed in an insulated tankers for delivery to processing plant. Okay, so if we talk about the collection center, then in a collection center the milk should be stored in a low temperature and it is stored in an insulated container so that the temperature should be maintained. Then after that it will transfer to the delivery processing plant. Under this handling and storage condition, the raw milk undergo a considerable chemical changes. Okay, so during the storage condition or during the handling condition, these raw milk are undergo a chemical changes, and these chemical changes are considerable due to the lipase activity and accelerate microbial growth. Okay, so the lipase activity are occur and it will accelerate the microbial growth. Suppose that if they during the milking 20 microorganisms are there, then it will reaches to a 20,000 microorganisms. Okay, it is an estimate value, but the microbial growth will accelerate it. But if we store it at a room, but if it is stored in a low temperature, that is the temperature range between 0 to 5 degrees Celsius, then we stop the growth of this microorganisms. Okay, but the lipase activities are occur. In temperature climate, normally milk is water cooled to as a low temperature as the cooling medium permit. The method of cooling and the temperature of the available water supply influence the final temperature. Okay, so the method of the cooling and even the temperature of the available water, they will what they will do, they will influence the final temperature. If we talk about the summer season, then it is possible that the dairy milk yield is stored around for 20 to 25 degrees Celsius for 14 to 18 hours. Now, where ambient temperature is frequently exceed 25 degrees Celsius. If the ambient temperature is exceed 25 degrees Celsius, then it should be stored between a 20 to 25 degrees Celsius and sometime 30 degrees Celsius. It is talk about the summer season. Then non-refrigerated milk is often collected twice daily because of the rapidity of the bacterial multiplication. Okay, if the milk are stored at a room temperature or a non-refrigerated condition, then in that condition the milking should be done twice a daily. Otherwise the bacterial multiplication is occur and it will lead to a consequent high risk of spoilage when milk is held at a such temperature for more than 6 hours. Okay, even 6 hours is more. If we left the milk for 2 hours at that particular temperature, then the microbial activity is occur and it will spoil the milk. On arrival at its destination, the canned milk is used immediately, cooled to GC and stored for more than 24 hours before processing or heat treated, and then cooled before a storage. Okay, so at that condition, when the milk are reaches to the destination, then the canned milk, the milk that is stored in a can, is used immediately. It is cooled at a GC and then again stored for no for not more than 24 hours before processing or heat treatment. Okay, so less than 24 hours the processing will start, heat treatment start, and then after heat treatment it should be cooled down and then stored. Now the next one is your bulk collection. Okay, so the next collection is your bulk collection. With the bulk collection, milk most common is refrigerated immediately after the production. Okay, if we talk about the bulk collection, then the name suggested that the milk should be stored in a bulk. So at that condition, after milking is done, immediately milk are refrigerated. Okay, and it is refrigerated either by means of an inline cooler followed by a storage in an insulated tank. Okay, so what should we do? We store it at an insulated tank where a inline cooler are there. The second one is your in a tank equipped with a refrigeration system, collection of milk which may be within hours. Okay, daily on alternate day, every third day or more rarely at longer interval is by means of insulated transport tanker, each picking up milk from several farm. Okay, so in that case, what they do, collection of the milk is done, suppose that it is done within an hour or daily, or even it is done an alternate day, every third day or more rarely at longer interval, and it is stored in an insulated transport tanker, and each picking up milk from a several farms. Suppose that A, B, C, three forms are there. Then the tanker should be take a milk from all these three firms. There is a risk that an undetected contaminated from one firm may turn the entire tanker load of milk. So here it is one risk, that suppose that if B B firm deliver a contaminated milk, contaminated milk, then it is a risk that these A and C both milking will be contaminated because of the same tank. Because if we take a contaminated milk, then it will contaminate all the milk. Milk haulers normally are authorized to refuse to collect milk from farm where the milk is tainted, appear to be abnormal, or is above a specific temperature, that is 7 degrees Celsius. Okay, so you can reject the milk if the if the milk that is collected from a farm is tainted, or sometimes it is appear abnormal, or it is stored above the specific temperature, that is 7 degrees Celsius. Bulk collection scheme include requirement concerning the design and performance of farm bulk tanks, the rate at which the milk is cooled, the maximum temperature at which it is stored, the frequency concentration collection, and the cleanness of the transport tanker. Okay, so here the bulk collection scheme include requirement concerning, like um, it included the design and performance of the farm bulk tank, even the rate at which the milk is cooled, and the maximum temperature at which the milk are stored, and the frequency of the collection, and the cleanness of the transport tanker, so that it will not contaminate the milk. On arrival its destination, if not used in immediately, then firm refrigerated milk is transferred to a insulated storage tank or silos, which will shown in a next figure. Suppose that this is your tanker from which milk are delivered to the dairy industry. Now here are insulated tanks are there, in which if the milk are not used immediately, then from the tanker this milk are transferred to the insulated tank, and here the milk are stored, and where it is stored at 3 to 5 degrees Celsius until heat treated for processing or manufacturing. Now this insulated tank have a temperature range between 3 to 5 degrees Celsius. Now here the milk are stored before the heat treatment are started or processing are started. In some countries the time delay before processing may be up to 4 days after the receipt of the milk. Okay, in some countries processing will be start after the 4 day after the receipt of milk. In the Netherlands, where some refrigerated milk is collected every third day, such milk, unless used immediately, is thermized, that is heated to 63 degrees Celsius for 15 seconds on arrival of the dairy. This process is sufficient to kill most of the psychotrophs, and after cooling to 5 to 6 degree Celsius, the milk is then can be stored for 2 to 3 days before use. A study of milk is thermalization was published by a Griffiths et al. 1986. Okay, if we talk about the Netherlands, in Netherlands, where our refrigerated milk are collected every third day. Okay, in that case, the milk are used immediately, it thermalized, that is it heated for 63 degrees Celsius for 15 seconds as it arrival in the dairy industry. Okay, so this temperature are sufficient for the killing of the most of the psychotrophs, and after heating it will cool between the range of the temperature, that is 5 to 6 degrees Celsius, and then it should be stored between 2 to 3 days before the use. But the temperature range should be 0 to 5 degrees Celsius during the storage time. Now the next one is your see. This is the diagram for the insulated storage silos are used to store a raw milk prior to a processing. The larger silos hold up to 30,000 gallon of milk, where the smaller one hold up to a 25-gallon, reproduced by a courtesy of the Milk Marketing Board, David Stowe. See, this is your insulated tank where the milk are stored, at least for milk are stored before the processing start, and here it is a tank where it is used for a transportation. Okay, this is also a tank. Okay, now the next one is your bacterial multiplication in stored milk. The temperature and duration of storage, the number and types of bacteria, and to a lesser extent, the natural inhibitory system in milk all influence the bacterial number increase that occur in a stored milk. Okay, so if we store a milk, then there are several factor which influence the bacterial number, like uh, the temperature and the duration of the storage. Suppose that we store the temperature at 13 degrees Celsius, sorry, it is 33 degrees Celsius for 3 hours, then the microbial count will be increases and it will spoil the whole milk. So the storage temperature and the duration of the storage will matter for a bacterial load. Now the second one is your the number and the types of the bacteria. It also influence the influence the bacterial load into a milk. Suppose that uh, some good bacteria are also there, then if the good bacteria are present in the milk, then it will not spoil the milk. And to lesser extent, the nature inhibitory system in a milk, these all influence the bacterial number increases that occur in a stored milk. Because of the wide variation in the initial microflora and the condition under which milk is stored, only generalization can be made concerning changes in a microflora occurring during the storage and transport. Now, the storage temperature probably is the most important factor in the microbial growth. Okay, so this storage temperature are very important. If we store the milk beyond the limit, then it will spoil the milk. Like figure 2.8 illustrate the likely effect of the temperature on milk of rather poor quality having a initial SPC of 50,000 CFU per ml. That is your CFU per ml. That means that CFU means that colony forming unit. Colony forming unit. Okay. Now the next one is your if the milk is not to be kept for more than about 12 hours, it is assumed that adverse effect will become appear when the SPC approaches 1 into 10 CFU per ml. Often milk is delivered in can is checked upon arrival for these effect, and if tented or if there is a evidence of an acidity above a specified limit, the milk is downgraded or the rejected. Now this is a graph between a total CFU per ml of a milk to the time in hour. Okay, so the effect of milk temperature on the increase in bacterial count in the raw milk having a initial SPC of 50,000 CFU per ml. Okay, so this is your time, that is your 0 hours, then 6, then 12, then 18, then 24, then 36, 48, 60 and 72. Okay, so this is the CFU count. Here you see that when the timer increases, then the bacterial load in a food will also increases. See, the graph will in a increasing order. Okay, now the next one is, now the spoilage organisms become a predominant at a temperature range of 25 to 30 degrees Celsius are mainly streptococci and coliform. Okay, so these two species, that is your streptococci and coliform, are become predominant at 25 to 30 degrees Celsius temperature, and they are what? They are a spoilage microorganism. Spoilage microorganism, that I already told you. This microorganisms are of two types. One is your pathogenic and another one is your spoilage. Okay, so the pathogenic microorganisms are capable of causing disease, capable of causing disease. Okay, but this spoilage microorganisms, they spoil the food product, spoil the food product. Okay, so this spoilage microorganism, that is your streptococci and coliform, they are predominant at the range of temperature 25 to 30 degrees Celsius, and they both are increase their number when the acidity of the milk will increase. However, unpleasant taste odor produced by a variation in the initial microflora can mask the clean acid odor. It is normally associated with the predominant streptococci population. Okay, if the streptococci population increases, then a unpleasant taste odor are produced, and this GNRs other than coliform and micrococci also will multiply unless or until any developed acidity become inhibitory to them. Okay, between 15 to 25 degrees Celsius, GNRS could outnumber streptococci. In general, the effects are similar to those at higher temperature, except that the adaptive phase of microbial growth is extended for several hours. Many species of GNRS can multiply in a milk and not produce any noticeable effect when their number reaches or exceed 1x. Okay, see, there are many species of the GNRS. Okay, what they do, they don't produce any noticeable activity in the food. Okay, unless their number reaches or exceed to a 1x. Okay, so they will spoil the food, but the they will spoil the milk, but the milk didn't show any characteristic of the spoilage. But when their number exceed from 1x, then it will show the noticeable effect. Thus, at a lower storage temperature, that is less than 10 degrees Celsius, the milk may appear normal for 2 to 3 days. That's why I just suggested that milk should be stored between 5 to 7 degrees Celsius. Okay, if we store milk at 5 to 7 degrees Celsius range, then it can be stored 2 to 3 days. Even thus, considerable bacteria growth has occurred. Refrigerated storage of raw milk. Refrigeration, see, when we store the food product in a refrigerated condition, that is at the range between 0 to 5 degrees Celsius, then it will inhibit the growth of the microorganism, inhibit the growth of microorganism. Okay, but their numbers are present. Suppose that this is your milk product. Before putting the milk into a refrigerated condition, the number of the microorganisms is 20. So when we store the milk at refrigerated condition, then the number will 20. It will not increases, but their growth will inhibit. Okay, this refrigeration by dealing bacterial multiplication can mask the effect of unsanitary production condition. In many dairy reasons, bulk collection of refrigerated milk account for most, if not all, of the milk produced. Because of this, the number and type of initial psychotrophic microflora present in a milk and their activities during our storage have become an increasingly important area of the concern. Alternate day collection is the most common practice worldwide. Okay, suppose that if a dairy industry collect a milk on Monday, then they will collect the next milk on Wednesday. Okay, so this is called what? This is called a alternate day collection, because four successive addition of milk are made to form bulk storage tank. About a quarter of the milk is 2 day old at a collection. There is a studies of alternate day collection between 1950 and 1970 which show no detrimental effect on milk bacteriological quality up to the point of leaving the farm, which provided the milk added to the tank after each milking was cooled repeatedly to a 50 degrees Celsius. There is a probably a slight increase in psychotropic number, but it would insignificant due to the dilution effect resulting from the repeated addition of fresh milk. However, the storage of the milk at 57 degrees Celsius of fresh milk with high psychotropic count most likely would lead to marked increase in bacterial count at the time of the collection, which result investigation of the bacteriological quality of alternative day collected milk were reviewed by Thomas and Roos in 1972. The initial total microbial count of a raw milk is of little value of predicting it count after refrigerated storage. Okay, so initial total microbial count of raw milk is of little value until it is stored in a refrigerated condition. Sample from a farm bulk tank taken shortly after the addition of a second milking were stored at a 5 degrees Celsius and SPC were determined at daily interval. The wide range of response is shown in the next table. Some samples show only small changes after 4 days, where others show a 3 to 8 fold increase in just 2 days. Okay, so if we talk about the different sample, then different sample have a different capability for the growth of the microorganism. Okay, suppose that we have a two sample, that is your A and B. Now A have a different quality and B have a different quality. Now maybe A show a some little responsible for the changes of this store storage. Suppose that A is stored for a 2 days. Now maybe the load of microorganisms is around 5000 CFU, but B when stored at for a 2 days, then it will show a microbial load around 10,000 CFU, just double. Okay, now, as might be expected, the fourth sample in which the bacterial multiplication has been most rapid at similar psychotropic and SPC after 3 days. Now, this table is SPC of milk which are stored at a 5 degrees Celsius in individual farm bulk tank. This is your A farm name. This is your 0 days. Load of microbial, then 2 days, then 3 days, then 4 days. Okay, see, if we talk about the farm A, then at 0 days the load of the SPC is around 5800. Now 2 days it decreases the value, that is 300. Then again their number is increases because the storage time is increases, it reaches to the 7900. Then on 4th day it reaches to the 14,000. Okay, now B at 0 days they have a macro SPC load around 14,000. Then 2 days around 10,000. Then at 3 days around 11,000. Now at 4 days around 70,000, and that's so on. Now the next one is your the effect of the refrigerated transport and subsequent storage. Because farm collection tankers are insulated, the temperature of the milk remained relatively unchanged from the temperature of the milk at the time of the pickup. Okay, so we all know that the milk are transported at an insulated tanker, due to which the temperature of the milk will not change when they are transported, and the temperature will remain same at the time of pickup temperature. The majority of the milk collected arrive at its destination at G degrees Celsius, and often they are no fasciculated for cooling it to a 14 degrees Celsius, because before it put into a storage silo. When it is stored, temperature ranging from 5 to 10 degrees Celsius, sample taken from both transport tanker and store silos to show increase in bacterial count. For milk which are collected daily, the result for a store sample have been initially psychotropic count is around 1 into 104 CFU per ml. are shown in the figure 2.9. Now suppose that and it is at which temperature? It is at 5 degrees 5 degrees Celsius temperature, and it is written in a cuisine etiol 1977, and this reported the count will increase around 1 into 10 to the power 6 CFU per ml in a 3 days, where other workers are also reported greater psychotropic increases. SPC of the compile of milk from storage silos were 1 into 10 to the power 7 CFU per ml after 1 day, and it incoming transfer tanker had SPC above 1 into 10 to the power minus 6 CFU per ml. For alternate day collection of the milk, according to the Muir et al. 1972, they recorded ranging of 10 to the power 4 to 5 into 10 to the power 6 CFU per ml for milk storage in a silos, which and it is stored around 24 hours at a plant. Now the next one, this is graph between the storage time days and the psychotrophs colony count per ml of the milk. Now, it is not clear to what extent contamination from transport tanker and associated houses pump meters and automatic sample increases the bacterial count of the milk. Because of the larger volume of the tank are involved, any significant increase in count per millimeter of milk are also indicate a heavy contamination of the milk which are come in contact with the surfaces. Some are increases in the count may be occur from the pumping process that disrupt clumps and change from of bacteria. However, there is no clear evidence concerning the extent of any such increase. One or two heavily contaminated individually farm supply well, of course, influence the bacterial content of the whole transport tanker load. For a milk that is more than around 2 hours in a train and has high content of the psychotrop, that is the multiplication of these microbes may occur with the milk temperature above a 4 degree Celsius. Now, type of the bacteria in store milk. Generally, milk are stored 2 to 3 days after the transfer from a transport tanker to a storage silos. The microflora of the milk are dominated by a psychotrop, where the thermoduric microflora have not changed significantly in composition. Now, spores of this psychotrop bacilli species can be present, but germination of these spores and outgrowth to any significant extent is unlikely. The predominant psychotropic genera and species will vary, but they are derived those present initially in the raw milk. Now, Pseudomonas species are the most frequently reported psychotrophs in store milk. This Pseudomonas species are the most common psychotrophs which are present in a store milk, with Pseudomonas jurensinus being the most common. Now, during the pasteurization, all psychotropic bacteria are destroyed in the vegetative state, but not those in a spore state. We all know that in pasteurization, what will happen? Only the pathogenic microorganisms are destroyed. Now, this pasteurization will destroy the psychotropic bacteria which they are in a vegetative state, but the spore forming are present. Pasteurization does not necessarily inactive the product of psychotrop metabolism or their enzyme. Okay, so this pasteurization does not have any impact on the psychotrophs who have a in a metabolism condition and also their enzyme. And there is an investigation done by a Schroeder et al. 1982, which indicate that the spoilage in a commercial pasteurized milk can be associated mainly with the GNRS and post-pasteurization contamination. This is contrast to laboratory pasteurized milk from the same source where the bacillus species were predominant microflora spoilage and no post-pasteurization contamination took place. Small-scale experiments show that the heat-resistant bacteria protease enzyme can affect adversely the product yield and quality, according to the Cuisines 1970 8. So this is all about the today's lecture. If you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is your Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website www.itlsacademy. Thank you for watching. Please like, share and subscribe our channel.